fluid. The only solution to that is to compact the actual arm itself, and then the part of your body which has more flexibility, that being your fingers, is then extended out, and it serves the purpose of a flipper now, or a fin. So these are pressures that occur over geologic time. Remember back to Lyell coming up with his concept of geologic time, that if you take the exact same DNA infrastructure and you expose that DNA to different environmental pressures, it will take the identical information and over time become something different. And so these arms here, all 6 of these arms, are considered homologies or homologous. And that's because they have the same, each of these bones has the same embryonic origin, but is now used for a completely different function. So the fingers of a bat and the front part up here, what are essentially the fingers of a bird, fingers of humans, the hooves of horses, these are all homologous because they all stem from the same ancestral DNA scaffold for how to make the front limb of a vertebrate animal. And this is also expressed in your embryonic development as well. So up here you have a chick embryo and a pig embryo, which tend to resemble themselves in their development. And it is only as that embryo continues to develop and as the DNA gets expressed slightly differently throughout that development that eventually you end up with a chick or a pig. But initially they start off with the exact same types of instructions, just read slightly differently because they have slight differences in their DNA construct. And guess what? Down here, this is you. You look very similar to a pig embryo and a chicken embryo when you are at the same level of embryonic development, somewhere around 3 weeks of development. And you'll notice in this lower picture here, there's a couple of things pointed out, but I want to point a few things out here too as well. You have what's known as a branchial arch. Branchial arch. Now this might be a little too small for you to read that, but that says branchial arch. And they're located about right there in the center. That means at 3 weeks of development, you actually have gill slits. That's right. Which again points to an ancestral relationship with other vertebrates who also exhibit gill slits, including chickens and pigs, and that's right, fish. Fish too. You also have a tail bud here at the end, okay, because we also possess the DNA for creation of a tail at some point in embryonic development. But in our case, that tail development does not continue on after a certain point. Okay, a couple more anatomical evidence pieces here. Vestigial structures. What is a vestigial structure? Vestigial means that it had a purpose at one point, but no longer does. So it was advantageous at one time, but nowadays it doesn't function in the same way. So a good example in humans include things like this, the tailbone. We don't have a tail anymore, and yet we still possess the bones that at one point were our tail. Our appendix doesn't seem to serve all that much of a function, but that's only true today. It actually served a much larger function back when our species consumed a whole lot more meat. Body hair, another good example of a vestigial structure. Okay, doesn't seem to serve an actual purpose today, but that's not to say that it didn't in the past. Other good examples outside of humans include what we mentioned before, the hind limbs in whales and snakes. That's right, there are some whales and that on occasion will have an error in DNA and they will actually produce little mini hind limbs. And also they possess a pelvis. And why do you need a pelvis if you don't have hind legs? Well, it is the remnants of the support system for hind legs. And this is true also in snakes, by the way. There are indeed certain snakes today that exhibit the remnants of hind limbs. And so you might ask yourself, well, you know, if there was a selective advantage to losing legs in whales, then why didn't it just continue in that loss and also lose the pelvis? Well, while there may have been a selective advantage to losing the hind legs, there is no additional benefit to continuing that loss and then removing a pelvis from the organism. It really came down to just streamlining. Once that body was streamlined, there was no additional benefit to losing more. But today we have molecular evidence as well. And this has caused our bag of evidence for evolution to just increase dramatically. And so let's talk about some things that point to the genetic relationship to all living beings on the planet. The reality is that if you are alive on this planet, that is, if you are a fungus, an amoeba, a rosebush, a blue whale, or any other thing, a sequoia tree, or a human being, you all operate on the same rules biochemically. That is to say, you all possess at least DNA or RNA, and that your DNA and your RNA always follow that ACTG rule, unless you're using RNA, in which it's ACUG. And it doesn't matter what you are. If you're a living organism, you use the exact same characters. The genetic code is totally universal. And we also know that no matter what you are, you all use 20 amino acids to construct all protein sequences. And it's the same 20 amino acids. So let me give you one line of biochemical evidence that's pretty telling. There is an enzyme in your body called cytochrome C. And what cytochrome C does is kind of irrelevant, but it is an important substance in your body. As you might imagine, since it's so important, the DNA sequence that might code for cytochrome C is going to have a strong pressure to remain the same, to not change very much. And because what happens if cytochrome C changes by even one amino acid? It ceases to function, right? So obviously, anybody that might have a mutation like that messes up their cytochrome C gene is not going to be around for very long. Certainly not long enough to reproduce. So, however, we do know that over long periods of time, you can get mutations that don't end up killing the organism. So, for example, in the cytochrome C gene, that is the actual DNA sequence that codes for cytochrome C, in humans versus monkeys, that difference is only a single base pair, a single DNA base pair, hinting at the fact that humans and all other monkeys at one point had a common ancestor. And you can continue this by looking at cytochrome C from other organisms, including pigs, ducks, turtles, fish, monkeys, and yeast. These are examples that we have here. And that chart, or the y-axis that you're looking at there, tells you how many amino acids different our cytochrome C gene is to their cytochrome C gene. And we are most closely related to monkeys, and the cytochrome C gene sequence kind of bears that out. We are similar to pigs in that we are distantly related to them. We are even more distantly related to ducks and even more distantly related to turtles, etc. And this is with one line of evidence, just one line of evidence, the cytochrome C gene. You can pick any other gene, like hemoglobin, for example. Let's pick hemoglobin, and it ends up showing a very similar story. In fact, you can take dozens of these and then take the trees that they create and overlay those trees, and they all end up looking very similar to this. And so this is corroborative evidence that agrees with a lot of the fossil evidence. We had mentioned early on in class that you don't lose DNA typically. Species that evolve over time, rather than losing DNA, they usually gain DNA. And the case in point here is that we brought up is this whole issue of whale hind legs. Whales today, modern whales, did not lose the DNA on how to make hind legs. Instead, they gained a gene that turns off leg production. So what happens when you knock that gene out? You can actually get whales with hind legs. It does happen on rare occasion. So let me ask you this question. How many nipples do humans normally have? What is the ancestral characteristic for that? Well, if you look back to other mammals, for example, cats, dogs, pigs, sheep, you name it, multiple nipples in females is actually the ancestral characteristic. It is what existed before just having two. So what happened in humans and other modern apes and monkeys? What happened to the instructions on writing multiple nipples? Well, we gained a gene which stopped nipple production at 2. So what happens when you knock that gene out? Well, you get people with multiple nipples. It happens. This is actually not very uncommon. And so on occasion, even we humans can revert back to an ancestral characteristic. So just in the case of whales, I wanted to show you a good example of that. Here are 2 examples. Here in the upper left, we have a humpback whale that is sporting hind legs. And this is an extremely rare thing. It says right here, this is the title of the paper that this picture came from. It's a remarkable case of external hind limbs in a humpback whale, and this was published in 1921. Absolutely amazing. Very, very rare. This colored picture here in the background is actually of a sperm whale, which is also sporting hind limbs. So it does happen. In 2006, a team of Japanese researchers actually found a dolphin that was sporting hind limbs. And so again, dolphins in general possess the capacity to make hind limbs. It's just that they possess a series of genes that turns off hind limb production. And but this one had that gene knocked out, and there it is, 2006. You can go and check it out. So really, this is the evidence that it happens, but the question becomes, how does it happen? And we are going to learn that in the next series of lectures. I will see you all in class.